Hello friends, warm welcome in this video, that is the second part of Kelvin cycle. We have already learned the first part of a Kelvin cycle in the earlier video, the link of which is given in description. Now we will study the stoichometry of Kelvin cycle. The Kelvin cycle regenerates biochemical components, that is the Kelvin cycle reactions regenerate the biochemical intermediates that are necessary to maintain the operation of this cycle. But more importantly, the rate of operation of the Kelvin cycle can be enhanced by increase in the concentration of its intermediates. That is, the cycle is autocatalytic. As a consequence, the Kelvin cycle has the metabolically desirable features, producing more substrate than consumed, as long as triphosphate is not being diverted elsewhere. The importance of this autocatalytic property is shown by experiments in which previously darkened leaves or isolated chloroplasts are eliminated. In such experiments, CO2 fixation starts only after a lag called the induction period, and the rate of photosynthesis increases with the time in the first few minutes after the onset of illumination. The increase in the rate of photosynthesis during the induction period is due in part to the activation of enzymes by light and in part to increase in the concentration of intermediates of the Kelvin cycle. Kelvin cycle stoichiometry shows that only 1/6 of the triphosphate is used for sucrose or starch. The synthesis of a carbohydrate starch sucrose provides a sink, ensuring an adequate flow of carbon atoms through the Kelvin cycle under conditions of a continuous CO2 uptake. An important feature of this cycle is its overall stoichiometry. At the onset of elimination, most of the triphosphates are drawn back into the cycle to facilitate the buildup of an adequate concentration of metabolites. When photosynthesis reaches a steady state, however, 5/6 of the triphosphates contributes to regeneration of ribulose 1,5-bisphosphate, and 1/6 is exported to the cytosol for the synthesis of sucrose or other metabolites that are converted to starch in the chloroplast. An input of energy provided by ATP and NADPH is required in order to keep the cycle functioning in the fixation of CO2. The calculation shows that in order to synthesize the equivalent of 1 molecule of hexose, 6 molecules of CO2 are fixed at the expense of 18 ATP and 12 NADPH. In other words, Kelvin cycle consumes 2 molecules of NADPH and 3 molecules of ATP for every molecule of CO2 fixed into carbohydrate. We can compute the maximal overall thermodynamic efficiency of photosynthesis if we know the energy content of a light, the minimum quantum requirement, moles of quanta absorbed per mole of CO2 fixed, and the energy stored in the mole of carbohydrate, that is hexose. Red light at a 680 nanometer contains 175 kilojoule, that is 42 kilocalorie per quantum mole of photons. The minimum quantum requirement is usually calculated to be 8 photons per molecule of CO2 fixed, although the number of 10 experimentally is 9 to 10. Therefore, the minimum light energy needed to reduce 6 moles of CO2 to a mole of hexose is approximately 6 into 8 into 175 kilojoule, that is equal to 8400 kilojoule or 2016 kilocalorie. However, a mole of hexose such as fructose yields only 2804 kilojoules, that is 673 kilocalorie when totally oxidized. Comparing 8400 and 2804 kilojoule, we see that the maximum overall thermodynamic efficiency of photosynthesis is about 33%. However, most of the unused light energy is lost in the generation of ATP and NADPH by light reactions rather than during operations of the Kelvin cycle. We can calculate the efficiency of a Kelvin cycle more directly by computing the changes in free energy associated with the hydrolysis of ATP and the oxidation of NADPH, which are 29 and 217 kilojoule, that is 7 and 52 kilocalorie per mole respectively. Kelvin cycles reactions, the synthesis of one molecule of a fructose 6-phosphate from 6 molecules of CO2 uses 12 NADPH and 18 ATP molecules. Therefore, the Calvin cycle consumes 12 into 217 plus 18 into 29 is equal to 3126 kilojoule, that is 750 kilocalorie in the form of NADPH and ATP, resulting in a thermodynamic efficiency close to 90%. The Calvin cycle does not occur in autotrophic cells. An examination of this calculation shows that the bulk of the energy required for conservation to CO2 to carbohydrate comes from NADPH, that is 2 mole of NADPH into 52 kilocalorie per mole is equal to 104 kilocalorie, but 3 mole ATP into 7 kilocalorie per mole is equal to 21 kilocalorie. Thus 83%, 104 of 125 kilocalorie of energy stored comes from reductant NADPH. The Calvin cycle does not occur in all autotrophic cells. Some anaerobic bacteria use other pathways for the autotrophic growth. The pyridoxine mediates synthesis of organic acids from acetyl and succinyl-CoA derivatives via a reversal of the citric acid cycle, the glycolate producing cycle, and the linear route, that is acetyl-CoA pathway of acetogenic methogenic bacteria. Thus, although the Calvin cycle is quantitatively the most important pathway of autotrophic CO2 fixation, other pathways are also important. Now we will study the regulation of the Calvin cycle. The high energy efficiency of the Calvin cycle indicates that some form of regulations ensures that all intermediates in the cycle are present at adequate concentration and that the cycle is turned off when it is not needed in the dark. In general, variations in the concentration or in the specific activity of the enzyme modulates catalytic rates, thereby adjusting the level metabolites in the cycle. Changes in the gene expression and protein biosynthesis regulate enzyme concentration. Post-translational modifications of proteins contribute to the regulation of the enzyme activity at the genetic level. The amount of each enzyme present in the chloroplast stroma is regulated by mechanisms that control expression of the nuclear and chloroplast genome. Short-term regulations of the Calvin cycle is achieved by several mechanisms that optimize the concentration of intermediates. This mechanism minimizes reactions operating in opposing directions which would waste resources. Two general mechanisms can change the kinetic properties of enzymes. First, the transformation of covalent bonds such as the reduction of disulfide and the carbomylation of amino group which generate chemically modified enzyme. And second, the modification of non-covalent interactions such as the binding of metabolites or changes in the composition of the cellular milieu, that is example pH. In addition, the binding of the enzymes to the alkaloid membranes enhances the efficiency of the Calvin cycle, thereby achieving a higher level of organization that favors the channeling and protection of substrates. Light dependent enzyme activation regulates the Calvin cycle. 5 light regulated enzymes operates in the Calvin cycle, that is Rubisco. Number 2, NADP dependent glyceraldehyde 3-phosphate dehydrogenase. Number 3, fructose 1,6-bisphosphate. Number 4, C-dihydrolase 1,7-bisphosphate. And number 5, ribulose 5-phosphate kinase. The last 4 enzymes contain one or more disulfide groups. Light controls the activity of these 4 enzymes via ferrodoxin-thyrodoxin system, a covalent thiol-based oxidation reduction mechanism identified by Bob Buchanan and colleagues. In the dark, these residues exist in the oxidized state, which renders the enzyme inactive or subactive. In the light, the disulfide group is reduced to the sulfhydryl state. This redox change leads to the activation of the enzyme, the resolution of the crystal structure of each member of the ferrodoxin-thyrodoxin system and of the target enzyme fructose 1,6-bisphosphate and NADP dependent malate dehydrogenase have provided valuable information about the mechanisms involved. This sulfhydryl, also called dithiol, signal of the regulatory protein thyrodoxin is transmitted to specific target enzyme, resulting in their activation. In some cases, such as fructose 1,6-bisphosphatase, the thyroidoxine linked activation is enhanced by effector, example fructose 1,6-bisphosphate substrate. Inactivation of the target enzymes observed upon darkening appears to take place by reversal of the reduction, that is the activation pathway. That is, oxygen converts the thyroidoxine and target enzymes from the reduced state to the oxidized state, and in so doing leads inactivation of the enzymes. The last four of the enzymes are regulated directly by thyroidoxine. The first, Rubisco, is regulated indirectly by thyroidoxine accessory enzyme Rubisco activase. The Rubisco activity increases in the light. The activity of Rubisco is also regulated by light, but the enzyme itself does not respond to thyroidoxine. George Lorimer and colleagues found that the Rubisco is activated when activator CO2, a different molecule from a substrate CO2 that becomes fixed, reacts slowly with an uncharged epsilon amino group of a lysine within the active site of the enzyme. The resulting carbamate derivative then rapidly binds Mg2 positive ions to yield the activated complex. Two protons are released during the formation of the ternary complex Rubisco CO2 Mg2 positive. So activation is promoted by increase in the both pH and Mg2 positive concentration. Thus the light dependent stromal changes in pH and Mg2 positive appear to facilitate the observed activation of a Rubisco by light. In the active state, Rubisco binds another molecule of CO2 which reacts with the 2,3 indiol from ribulose 1,5-bisphosphate, yielding 2-carboxy 3-keto ribitol 1,5-bisphosphate. The extreme instability of the latter intermediate leads to the cleavage of the bond that link carbons 2 and 3 of a ribulose 1,5-bisphosphate, and as a consequence Rubisco releases two molecules of a 3-phosphoglycerate. The binding of sugar phosphates such as ribulose 1,5-bisphosphate to Rubisco prevents carbamylation. The sugar phosphate can be removed by the enzyme Rubisco activase in a reaction that requires ATP. The primary role of a Rubisco activase is to accelerate the release of bound sugar phosphate, thus preparing Rubisco for carbamylation. Rubisco is also regulated by natural sugar phosphate, carboxy-arabinitol 1-phosphate, that closely resembles the 6-carbon transition intermediates of the carboxylation reaction. This inhibitor is present at low concentration in leaves of many species and at a high concentration in leaves of legumes such as soybean and bean. Carboxy-arabinitol 1-phosphate binds to Rubisco at night and is removed by the action of Rubisco activase in the morning when photon flux density increases. The recent work has shown that in some plants, Rubisco activase is regulated by the thyroidoxine system. In addition to connecting thyroidoxine to all 5 regulatory enzymes of a Calvin cycle, the finding provides a new mechanism for linking light to the regulation of enzyme activity. Light-dependent ion movements regulate Calvin cycle enzymes. Light causes reversible ion changes in the stroma that influences the activity of Rubisco and other chloroplast enzymes. Upon illumination, protons are pumped from the stroma into the lumen of the thylakoid. The proton efflux is coupled to Mg2 positive uptake into the stroma. These ion fluxes decreases the stromal concentration of H positive pH 7 to 8 and increasing that of Mg2 positive. These changes in the ionic composition of the chloroplast stroma are reversed upon darkening. Several Calvin cycle enzymes, Rubisco fructose 1,6-bisphosphatase, CDF-2-lose 1,7-bisphosphatase, and ribulose 5-phosphate kinase are more active at pH 8 than at pH 7 and require Mg2 positive as a cofactor for a catalysis. Hence, these light-dependent ion fluxes enhances the activity of key enzymes of the Calvin cycle. Light-dependent membrane transport regulates the Calvin cycle. The rate at which carbon is exported from the chloroplast plays a role in regulation of the Calvin cycle. Carbon is exported as a triphosphate in exchange for orthophosphate via the phosphate translocator in the inner membrane of the chloroplast envelope. To ensure continued operation of a Calvin cycle, at least 5/6 of the triphosphate must be recycled. Thus, a most 1/6 can be exported for sucrose synthesis in the cytosol or diverted to starch synthesis within the chloroplast. The regulation of this aspect of photosynthetic carbon metabolism. Thank you.